Welcome. In this lecture, we will discuss some of the important questions from botany. And important to note, many of the questions were from genetics, biotechnology, a very important topic in light of the RT-PCR. Lots of questions were there from PCR, that is polymerase chain reaction. But however, this topic again becomes important for your 2022 examination. Now, in this video, we have covered the RT-PCR in detail and how does the PCR work, which is the polymerase chain reaction. So in the first round, it is the denaturation followed by annealing and extension. The details you can check the video. A very important concept. Now similar concept on the same topic was asked, that which is not an application of PCR. So here we understand that with the help of polymerase chain reactions, when you are multiplying, you can do a molecular diagnosis, do amplification of the gene, and diagnose any kind of mutations in the genetic network. However, it is not used for the purpose of purification. So 1, 2 and 3 are the correct option. And note, this question asks about not. So read the question very carefully while you are answering the topics. Now, this is a question of match list. A question which has been which you have been studying since class 7 or 8 in your NCRTs. So even if you are able to match 1 or 2, you should be able to complete the remaining ones. So that is one of the benefits of match list questions. Try to match 1 or 2 in which you are 100% confident. Then even if you are marking, let's say you have choices A, B, C and D. A and B have the first match which is correct from your side as well. C and D are not the options. Then even if you are not able to get to the exact right answer, you can actually be very much close to the correct answer, even if you are just marking it by chance there. So the idea is, which cell have active cell division capabilities? We have understood meristematic tissues, and they are of 3 types: apical, which grows on the tip, lateral, and intercalary between the nodes. So this type of cells are called as meristematic tissue. So the A goes with meristematic tissue. The next is the dead cells. Now dead cells are the sclereids, and these have thickened walls and narrow lumen. So again, a very direct match that you can see here. Now which tissue have different types of cells? Vascular tissue have xylem and phloem. Then xylem again you have xylem parenchyma. Phloem again you have phloem fibers, phloem parenchyma, and so on. So tissues which have different types of cells are what are called as vascular tissues. Now what remains is the tissue having all cells similar in structure and function are the simple tissues. So again, a very direct question I could say which was asked here. Here we have how the transfer of pollen grains takes place. Now the transfer of pollen grains from the anthers of one plant to the stigma of a different plant during pollination, and this brings in definitely a different gene to the stigma. So anther from one plant is going to the stigma of the another plant, and this anther goes to the stigma of the another plant, and this process is known as what? It is known as xenogamy. Now if it is to the other plant, it is known as xenogamy. Xenogamy is also known as allogamy, or what we call as cross pollination in very simple terms. Okay, then we do have other types of pollination methods which are mentioned here. All these remaining 3 are the types of self pollination. Now cleistogamy occurs when the flower never opens and the pollens are deposited on the stigma of the same flower. So this flower does not open for others. The pollens are deposited on the stigma of the same flower. Cleisto, the word itself means what? Closed. So this is a kind of self-pollination. Genitogamy and chasmogamy are again the kinds of self-pollination which is done. Okay, in genitogamy, what happens is the pollen grain from one flower is transferred to the another flower of the same plant. So this plant is there. On this plant, this is one flower and this is another flower. So from one flower of the plant to another flower of the same plant, it is known as genitogamy. Okay, so that's how it is called as. So we have talked about some of the major forms here, and understanding this question is that it is a very basic question usually asked. Carrageen, a very direct question usually asked. It is obtained from where? It is obtained from red algae. Similarly, we have another question that was, I believe, from brown algae. So direct questions again. Now meiosis, in the stages of meiosis, at which stage centromere divides? So centromere divides at anaphase 2. A very direct question. We have covered this in our class on stages of meiosis 1 and 2. So in this class we have covered significantly only meiosis. In the other class we have covered the difference between mitosis and meiosis, so refer that. Now this is a very important section, very scoring section. Ecology and ecosystem comprises around 8 to 7 questions in the exam every year. One of the most simplest section and the most scoring section. The energy pyramid is always upright is correct. You have to find out which of the following is not correct. Okay, the pyramid of number in a grassland ecosystem is upright because you have highest number of grass followed by less number of deer followed by very few lions, so the pyramid becomes upright, so again correct. The pyramid of biomass in the sea is inverted and the pyramid of biomass in the sea is upright. Now note, one of these could be the correct sentences. So definitely either 2 is wrong or 3 is wrong, but we know the biomass in the sea is inverted. Why? Because you have lots and lots of fish but their weight is very less. As we move to the top hierarchy, whale, the weight becomes very high, so this pyramid gets inverted. So second is the right option, third is the incorrect option, and this we have discussed in our class in detail on the following link. The next is diadelphous stamens. Now diadelphous stamens are where the stamen is actually seen into 2 sections. If it is fused into one, we call it as monodelphous. Now dia means 2, mono means 1. So good example where we have diadelphous stamen is pea. So pea is a good example for diadelphous crop, usually seen in the Fabaceae species. So that's where we see the diadelphous stamen. A very important question, and this is also important in line of the Mendel's experiment. So while reading the genetics again, we have talked about this. The next is amensalism. Now mutualism, neutralism, commensalism, parasitism, predation, amensalism and competition. All of these terms are important. Amensalism is a process where one of the species is harmed, the other is unaffected, and we call this process as amensalism. So in this video we have covered all the 7 types along with their examples, a very important topic, so don't miss it. The next is we have the various phases and which phase relates to what. So again a very direct question of cell cycle interphase, the various phases under interphase, so we have covered that separately. Now what happens in the S phase? In the S phase there is replication of the DNA. So under the S phase what happens is DNA replication takes place. In the G1 phase you have the initiation of the replication of the DNA and it is the gap between the mitosis process, so we call it as G1 phase. D2 phase is the phase where we have the synthesis of the protein that takes place, and quiescent, as the name suggests, this is the inactive. So wherever you have the word quiescent related to inactive, and you have the right match here. So as you can see, there were numerous questions which pertain to match list. The questions from genetics were significantly higher. The section from genetics, biotechnology, even plant classification, we had few questions, but the questions were relatively simple. As we said, one of the questions was from red algae, the other question was from brown algae, and also the scientific names of some of the species were asked. So those are some of the important topics that you must cover. As always, we have seen 100% sure questions from the section on meiosis and mitosis. Very important to cover. Interphase, cell cycle. Then we have the differences between mitosis and meiosis, extremely important section, must to cover. Under genetics, hybrids and cross, F1, F2, very important, must to cover. Biotechnology, polymerase chain reaction, DNA polymerase, RNA synthesis, transcription, translation. So all of those become extremely important in the section on biotechnology. So this was just a quick summary of some of the important questions, what you need to solve, how you need to proceed, and for more practice, refer also the website which is FlexiPrep. On FlexiPrep, we have covered some of the solved questions for your convenience, so you can also check those for free. Thank you very much for joining.